Hello dear students, in this video we are going to continue with our gastrointestinal system only. So we were doing the part 2. In the part 2, the first video we have done about the GAT hormones. Of course, GAT hormones is very important, and in some of the institutions, the next topic, today's topic, that is the physiology of digestion and absorption of nutrients, this is also important. This topic is closely associated with biochemistry. So if you are studying from biochemistry also, it's good enough. So either of the places you have to study all this digestion and absorption of nutrients. So first, coming to the topic, the digestion of absorption of nutrients, we will be discussing them under 3 headings. One is the carbohydrates. So today our discussion will be on the digestion of and absorption of the carbohydrates. In the next coming videos, we will be discussing about the protein digestion and absorption, followed by the fat digestion and absorption. These are the 3 major important digestion and absorption you should know. So first we will be discussing about the carbohydrates, because that is a major source of all the energy that we get in. So carbohydrates. So the carbohydrate digestion primarily involves digesting of the 3 substances. One is the polysaccharides. The commonest carbohydrate that we intake is the polysaccharides, that is nothing but the starches. So they are also referred as starches. Coming to the disaccharides, the substances are like sucrose and lactose. These are the substances which come under disaccharides. Coming to the monosaccharide, which is the smallest molecule, the glucose. So these are the major important carbohydrates which we should be undergoing the digestion has to happen and followed by the reabsorption. So let's first understand their structure and how those molecules has to be broken down to form the monosaccharide so that reabsorption can happen. First, when we ingest substances, majority of them are made up of the glucose molecules and along with some linkages. So first example, the glucose molecules, when they are having the linkages like the alpha-1,4 bond, they are called as amylose. Next, we have another substances wherein they have the side chains. So this is going in a straight line. This is called as linear direction. So this one we can see here, it is linear in nature. Whereas the next one that we are going to see, it is having some side bonds. And these linkages are called as alpha-1,6 bonds. So the side linkages are also present. The commonest enzyme, for example, that comes from the salivary gland, the salivary amylase or the pancreatic amylase, all those amylases, the amylases can break only this linear bonds. As you can see here, in the linear fashion, it can break it. But wherever the side bonds are there or the branched ones are there, wherever the alpha-1,6 bonds are there, they cannot break them. So for that, we will be needing some more additional substances. So when these amylases act in, we will land up with end products which are nothing but the maltose, maltotriose because of 3 molecules and glucose oligomers followed by alpha-dextrin. So now we have to further break down this maltose, maltotriose and alpha-dextrins to smaller molecules. So the maltose and maltotriose, as you can see here, they will undergo much more molecular division with the help of the enzymes glucoamylase, sucrase and isomaltase. So this isomaltase is very important for breaking them and then bringing down the final molecules, which could be a glucose or some other molecules. Then alpha limit dextrins, the ones which is having the side bonds, the side linkages or the band linkages. For that we need additional steps. First, the glucoamylases will come into action. Some of the glucose molecules can be broken down. After that, our favorite enzyme, that is the isomaltase, will come because the side chain breakage is majorly done by the isomaltase. And again, the same glucoamylase, sucrose and isomaltase will form them and break them into the final molecules or the smaller molecules. Because we have transporters only for the smaller molecules which can be reabsorbed into the intestinal lumen. That's why we have to break these bigger molecules. So let's see. The sources, 3 major sources of any kind of food will be the starches, the lactose and the sucrose. Whenever it acts upon the saliva, the enzyme is salivary amylase, which is also called as ptyalin. It is not a major enzyme because it can cause some amount of digestion, but as soon as this reaches the GAT, this salivary amylase is inactivated by the increased HCl secretion, or we can say that whenever there is a decrease in pH, they can get deactivated because they do not function in low pH values. But the pancreatic amylase is one of the major important enzyme which is going to break down them. And these starches are generally broken down into the substance called maltose. Just now we saw maltose can be further broken down by the intestinal enzymes which are the maltase. And finally, Otherwise, isomaltase also we can say isomaltase. And finally, we will land up with the smallest molecule, that is the glucose. In a similar manner, the lactose will also be acted upon by the intestinal enzyme, that is lactase. This lactase is very, very important. We will see an applied aspect also. And finally, it will break into another form of glucose. So, lactase further broken down into glucose and galactose. From maltose also we have derived the glucose, but lactose we derive glucose as well as galactose. In a similar fashion, the sucrose is further broken down with the help of the intestinal enzyme sucrase into glucose and fructose. If you closely observe, there are three end products I am getting. The first one is the glucose, which is very, very important. Next, we have the galactose and the fructose. So, these are the three end products we are getting. So, the end products I will just name it glucose and galactose and fructose. So, now we have all these three substances. Because we had the bigger molecules, all of them are broken. Now, we have the end products. These end products has to be reabsorbed into the intestinal lumen. So, any reabsorption, first there will be a lumen. Suppose for example, this is the intestine. So, this is the lumen. So, the food is coming here. Now, we have broken them. From the lumen, it has to be transported, and whatever energy substances we are getting in, ultimately they have to reach the blood vessel. So, this part or the apical side is called as the luminal side because the lumen is there. So, this is called as the luminal border. Whereas the other side which is facing towards the blood vessel, that is called as basolateral side. You can remember it. The one which is facing the lumen is luminal side. The other one which is facing the blood vessel B for B, basolateral side. So, that is what this diagram is depicted. Here there is a blood vessel shown, and blood vessel side, whatever the cell is facing, that is the basolateral side. The cells, this side will be called as the luminal side. So, this is very, very important because time and again we will be asking about what is the particular transporter present in the apical side or the luminal side, what is the transporter present in the basolateral side. Because when we are going to give drugs to the patient or when you are going to read pharmacology in future also, you should understand which transporter you are going to block and where it is present. So, that is why this knowledge is important. And this glucose and galactose, both of them can be transported along with the sodium. So, both of them can be transported along with the sodium. With the help of a transporter, I have named this transporter here, that is nothing but sodium glucose transporter. In general physiology also we have studied this transporter. So, I will just write it fully, sodium glucose co-transporter. They are going to go together in the same direction, that is why it is called co-transporter. But this co-transporter is a secondary active transport. If it is a secondary active transport, somebody has to put the energy. That is done by the primary active transport, which is present in the basolateral side. That is nothing but our famous sodium potassium ATPase pump. So, that is the one which is going to provide the energy to this pump indirectly. Next, for the fructose we have another transporter which is called as the GLUT5 transporter. GLUT5. So, the number B labeled here is GLUT5. This SGLT, now we are studying the gastrointestinal system. Again, in renal system also we will study the same SGLT. But here the variant is type 1. Type 1. In the kidney, there is type 2. I will just write it down here also so that when we read kidney, it will be more useful. So, how many kidneys we have? That is the easy mnemonic to remember. So, we have 2 kidneys. So, it is SGLT2 in the kidneys. How many gastrointestinal tract we have? The gastrointestinal tract is 1 tract. So, this is has to be. And they ask these questions in INICT, NEET and other entrance examinations also. So, pay attention to all these important transporters. So, SGLT1, we have it. Now, if I want to give a drug to a diabetic patient to reduce their glucose levels or to send the excess glucose out of the body, I should block the SGLT2 only, because if I block this, the glucose will go into the urine, which is good for the patient because the patient is having high glucose in the blood. But if I just block a blocker, which is just SGLT, which blocks both SGLT1 and 2, what will happen to the patient? If I block this SGLT1 by mistake, the glucose will not enter into the body, the patient will die out of hypoglycemia. That's why they have designed the drug in such a way that it will block only the SGLT2 in the renal system and it can be excreted out in the urine. Okay. So, for a first year, it's a little bit more of information, but overall in future, you will apply all these things. Next, fructose, how will you remember this transporter? F, you can easily remember as F is for 5. So, the transporter is GLUT5, okay, GLUT5. And both of them will enter into the cell, that is the GI cell, and after that they will be transported into the basolateral side. Most of the times it goes into the basolateral side with the help of a GLUT2 transporter. Here GLUT5 and GLUT2, all of them are facilitated diffusion. So, these two are nothing but facilitated diffusion, So, this is all about the absorption aspects. Now, coming to the applied aspects, our famous drug, whenever any patient is having diarrhea, what we are going to give them? We are going to give them ORS. If you closely observe this oral rehydration solution, this is nothing but oral rehydration solution. It will have two important elements. One is the sodium, another one is the glucose. So, when a patient is having diarrhea, he will have electrolyte loss also and energy losses also. But when I give a substance, I should give both sodium and glucose, because when I give them in this combination only, I can activate the transporter, that is SGLT1, which is predominantly present in the GAT. This can be activated only when both are present. So, that's why our parents, they have an inherent knowledge, like most of the mothers. What they do is, whenever a baby is having diarrhea, they will mix both salt as well as sugar and make a homemade preparation of the ORS and give them for the diarrhea. That is because the logic because is here is they both, only the presence of both of them will activate the SGLT1. So, ORS is one of the important applied aspects. Never give a person of diarrhea, when you give lemon juice also, don't give just with sugar. You should always put in a salt so that the reabsorption is better. Coming to another one, lactose intolerance. Just now we saw, to break it down, we need the enzyme, that is lactase. If lactase is not available, what is going to happen? Lactase, their levels are very, very high in newborns. Whenever a newborn is born or the baby is having a young age, we can say, the lactase level is very, very high. But as and when the person goes older, the lactase level starts to decrease. That's why you will commonly hear in people who are above 30s and 35s, whenever they have some milk products, whenever they have a milk product, and milk products will have a lot of lactose in it, and now they don't have the lactase in them. So, what will happen is they will have intolerance to this lactose, that is called as lactose intolerance. This is an osmotically active substance. So, what it does is it is going to reabsorb the water. So, now more and more water will be present in the lumen and it will cause something called as bloating. So, that is why whenever they people with lactose intolerance, when they take many kind of milk products, this is going to happen. So, what is the solution? The best solution is avoid milk products. But the problem is many a times when you try to avoid milk products, you have to give up a lot of good substances, good things like you enjoy. Like for example, people who love ice creams, it will be really difficult for them to give away with it. So, there are other treatments also, like artificially available lactase is also available, but best treatment is to avoid those substances or take in a lactose free substances. Suppose for example, nowadays we are getting lactose free milk, lactose free ice creams and so on and so forth. So, that can be taken. So, this brings us to the end of this video. So, in the next video we will be discussing about the digestion of the proteins and followed by the fats. So, thank you for watching. If you have any doubts, please drop it in the comment section. I will be happy to help you. Thank you so much.